think very few of us are actually going in for testing. So if anybody has symptoms, they're self-isolating themselves. So, I mean, the public has, I think, kind of just accepted that it's something that's going to happen along with so many other diseases that are in the pipeline or going on in India, something like that.
That's good.
Yeah,
that's
true.
You could hear me well?
Yes, we can hear you, sir.
Wonderful. दूसरी क्लास पहले छूट गई थी
शत शत नमन हमारा इस ज्ञान ज्योत को शत शत नमन हमारा इस ज्ञान ज्योत को वंदन प्रणत हमारा इस शुभ परिसर को शत शत नमन हमारा इस ज्ञान गीत जिंदगी के सीखे जिसके आंचल में मुनि मंत्र सिद्धांत जहाँ में ज्ञान कला उड़ती नई क्षितिज बहुरंगी जहान अंतर उड़ती नई क्षितिज बहुरंगी जहान अंतर मिलती विविध दृष्टियाँ अनगिनत विषयों पर विनत हमारा मस्तक इस विद्यामंदिर को विनत हमारा मस्तक इस विद्यामंदिर को वंदन व्रत हमारा इस शुभ परिसर को शत शत नमन हमारा इस ज्ञान स्रोत को भाव बुद्धि मन होते विकसित जहाँ हमारे मूल सभ्यता संस्कृति के हो मुखरित सारे सीखा पाठ जहाँ हमने समता ममता का सीखा पाठ जहाँ हमने समता ममता का सहिष्णुता सहयोग वेदना और विवेक का अर्पित पुष्प हमारे इस कर्म भूमि को अर्पित पुष्प हमारे इस कर्म भूमि को वंदन व्रत हमारा इस शुभ परिसर को शत शत नमन हमारा इस ज्ञान स्रोत को
morning. Greetings to one and all who have connected with us for this Centenary lecture series. On behalf of our Honorable Vice Chancellor Professor Alok Rai, our Head of the Department Professor Sudhir Pawar, faculty members and my own behalf, I welcome you all in the Centenary lecture series. This year, our University of Lucknow has completed its 100 glorious years of light and learning. It is a great privilege for us to listen such esteemed personalities who have won accolades in their respective fields. Before we begin our Centenary lecture series, I would like to draw your attention that our Department of Zoology is also entering in its 100th year. So today's eminent speaker with us is Dr. Samer Hattar. He is a chronobiologist and a leader in the field of non-image forming photoreception. He is currently the chief of the section on light and circadian rhythms in the National Institute of Mental Health, part of the National Institutes of Health. He received his PhD from the University of Houston in Texas under the mentorship of Arnold Eske. From 2000 to 2004, Dr. Hattar was a postdoctoral fellow at Johns Hopkins University School of Medicine and the Howard Hughes Medical Institute. In 2004, he joined the faculty of the biology department at Johns Hopkins University with a joint appointment in the Department of Neuroscience at the Johns Hopkins University School of Medicine. In 2017, Dr. Hattar joined the faculty of the Intramural Research Program at the National Institute of Mental Health, and his research focuses on light effects on circadian rhythms, sleep, mood, and learning. His biography is full of bests. He has immensely contributed in the field and has received several awards and honors. He has also published his research in the best journals of the field, which are peer-reviewed and have a high impact. To name a few, his research has been published in journals like Nature, Science, Journal of Comparative Neurology, Nature Reviews Neuroscience, PNAS, Journal of Neuroscience, Chronobiology International, Cell Reports, Current Biology, and so the list is long. So with this important and interesting information, I invite Dr. Hattar to deliver his lecture on diversity of light effects in animal behaviors. So the stage is yours, Dr. Hattar. Can you hear me?
Yes, I finally was able to unblock the mute. Thank you, Professor Malik, for inviting me, and thanks for Professors Ray and Kumar for leading this very exciting occasion of 100 years of a very esteemed university that I've read about, that produced presidents in India, important scientists, and a lot of people that have benefited from an amazing educational system. So I'm gonna share my slides with you. And today I'm gonna tell you about how light affects animal behavior, and I'm really excited to talk to a zoology department because I would love to get any suggestions, any insights after my talk about what you think the direction or the future holds for us. So before I start my talk, I would like to acknowledge the people who helped me do the work. These are two postdocs currently working under the supervision of Dr. Haichin Zhao at Johns Hopkins University. And at Johns Hopkins University, as Professor Malik said, we were part of the three mouse trial lab where myself, Haichin Zhao, and Rachel Grovilla, who happens to be my wife, we were sharing resources and reagents, and students and postdocs and undergrads, which was a really exciting environment for young faculty to start in. And as mentioned, I have moved from Johns Hopkins to the National Institute of Mental Health at the National Institutes of Health, and this is my current lab members, and I'm really excited about telling you about some of the work that we have been able to achieve in all of these years. So light is important for you to enjoy this image here. So light goes through the pupil and then hits the back of your eye where the retina, the light sensitive part of the eye is located, and then through the optic nerve which connects the eye to the brain. This information reflected from this image goes to regions in the brain such as the lateral geniculate nucleus, specifically in mice the dorsal portion of that, as well other brain regions, and then to the visual cortex to allow you to consciously see this image. So although this image is two-dimensional, what you could glean from this looking at this image that there are actually cliffs, there's waterfalls, there's even a rainbow. But light also is important for functions that we collectively call non-image forming functions, and these include simple reflexive behaviors such as the pupil constriction, where your pupil constricts in response to light. And this actually, although is a very simple behavior and can be literally directed by a simple light sensitive photopigment in the muscle, what you could see is that somebody's telling me they can't see my slides. Can people see my slides? Can you see my slides?
Yes, they are visible.
Oh, okay. Because Ruchi was telling me she can't see my slides.
Yeah, we can see it.
You could see the slides. Okay.
Yeah.
So as I was saying, that there is a non-image forming functions for the eye, that a pupil constriction, for example, you could have a photopigment in the pupil muscle that could drive the responses directly in the eye. But remarkably, this very simple reflexive behavior requires the retina and requires at least multiple synapses from the retina to the level protective nucleus and then back to the eye to drive this pupil constriction. And finally, there are functions to light that are also very important for our sleep-wake cycle, for our daily behaviors, and these were collectively referred to as circadian photoentrainment, where you adjust your internal circadian time to an exact 24-hour time. I'll be talking a lot more about this, so you shouldn't worry about it right now. And this is usually driven by two brain regions that we're known of are the suprachiasmatic nucleus, or SCN, and the intergeniculate nucleus. I'm sorry, Ruchi still says that maybe in the YouTube they can't see my slides, but maybe you could see it here, but maybe in the YouTube channels people can't see my slides.
Yeah, we are trying. There is some problem, but the person is working on it.
So I'm just gonna continue then. Okay, wonderful. So why is it?
Just a second, Samar. I'm sorry to interrupt. Anuriti, can you just tell what he is doing? What is to be done?
Good morning, sir. Sir, you have to keep your slides open and not the Gmeet screen. Then we can see your presentation.
You're putting the Google Meet screen open. Please do not keep that open. Just the your presentation slide.
Just what about now? Like this?
Anuriti? Yeah, I think this will work. Anuriti, will it?
Do they see my slides now?
Anuriti, can you sort it out?
Ma'am, sir, your slide is not visible in the Gmeet screen.
How do you suggest he does that?
Should we go for full screen?
You can, sir, you can only open your presentation PPT and let it open. We will see that on our screen.
No, you're not here. Sama, can you just stop presenting for a second?
Sure.
Can you stop presenting? Yeah.
Just stop presentation.
Yes. And yeah. And can you now?
Okay, stop presenting. Okay, cool.
Yeah. And now you have your presentation open in the background, right?
Yes.
Okay. So now, if you click present now and you select a window, and then you just open your. If you click present now, you will see three options which say your entire screen, a window, and a tab.
Okay, let's see. Present now. It says Your entire screen?
So
A window.
on the top.
And then you see the second option is a window. So you need to click a window.
On mine, unfortunately, it only says your entire screen. There is nothing else.
Okay, then that is what so you need to click your entire screen. That is why it's creating the problem. So it's not showing the other two options.
No, it's not. So this is now, I said my entire screen.
That is why we have the problem. So can you open your...
Do you know if this is maybe I'm using Centenary. Maybe I should use the Firefox, for example.
Yeah.
Or you can use Edge. I think Edge everybody will be having. I think it won't be a problem. You can try with the other one. So the three ones are coming. Go to the program now and just
Sir, if you want, you can try one more time. You can just minimize this Gmeet screen and open your presentation. I think that will that might solve the problem.
Just minimize this and open the presentation. Do you see it now?
Yes, sir. Yes, sir. We can see it. You can put it in full screen. So.
What about now?
Yes, sir. The presentation is visible.
Oh, wonderful, wonderful. I'm so happy. So, I'm sorry we wasted a lot of time, but maybe just quickly, because you didn't see the first slide. These are the people who are working with me. We're part of the Mouse Tri Lab, as I said, and this is my current lab at the NIMH. And the eye is responsible for detecting conscious images and also for functions that are non-image dependent and are really subconscious in nature. And one of the important functions that light does is that it adjusts our timekeeping mechanism to match the solar day. And you could observe this in a human very nicely in our sleep-wake cycle, as you could see here across several days, as well as in our temperature. Although we're supposed to be isotherms, we have very predictable variation in our body temperature that happens in the daily cycle. And what's really amazing for me and what really excites me about this field is the fact that even our cognitive abilities have daily variations, where we have different times of the day that we're able to compute mathematical equations better than others. So although every person is different, unless you're an identical twin, every single individual is different at different times of the day. So you're not the same person in the morning, at noon, or early evening hour. You're not only changing physiologically, but also hormonally and genetically, and so on and so forth. So it's important to understand how these variations in the time of day affect the physiology of animals. So how do we model this in the lab? The way we model this is actually quite simple. We put a mouse in a cage with a wheel. Every time the mice run on the wheel, we record the wheel revolutions. And then we put this single animal for several days in different light-dark conditions. So for example, in the first 12 days, these animals are maintained under 12 hours of light, 12 hours of dark. And because they are nocturnal animals, they can confine their activity to the dark portion of the cycle. And what you could see here is that mice run tremendously, a huge amount on the wheel at night. So for example, if you calculate the amount of running they do in a single night, it comes out to be us running at 4 to 6 miles on a treadmill every night. And what you could see is that even when you put these animals under constant conditions, in this situation, constant darkness, these animals still have a circadian clock, but it's not exactly 24 hours anymore. In the case of this mouse, it's 23.6 hours. If I put you in as a human in these conditions, you'll have a longer than 24-hour period, because the human circadian clock runs up around 24.2 hours. You could actually jet lag these animals, so you could bring them, for example, from New York to Amsterdam or Amsterdam to New York. And what you could see, it takes them 6 days to adjust to a 6-hour shift. So you could see when you shift them where the sun comes on earlier, you will actually have 6 days to adjust to the new cycle. And the same thing happens when you delay the onset of the morning. So as I told you, the eye is responsible for detecting functions for both image and non-image forming functions. And the question is, which photoreceptors in the retina are responsible for this? And the mammalian retina have been studied for hundreds of years, and people have understood the retina at quite high levels when circadian photoentrainment was started to become popular. And the general understanding is that the retina is inverted in our eyes. So if you take your eye and you look through the pupil, the first layer you see is the retinal ganglion cells. These are the cells that have axons that form the optic nerve that I told you connect the eye to the brain. So light has to cross all these retinal neurons before it hits the photosensitive parts of rods and cones that have the famous opsins, rhodopsins, and conopsins. These rhodopsins and conopsins take the light energy in the form of photons and through a phototransduction pathway change that light energy into an electrical signal that the neurons can understand and send back through the retina to the ganglion cells, which send this information to the brain as electrical impulses. Now, remarkably, people have thought for many, many years that only rods and cones are the photoreceptors in the retina. The rods is what allows you to see in very dim light conditions, and cones is what allows you to see in brighter light conditions and allow you to see color. However, in 1999, Russell Foster and colleagues did experiments that I thought were really fascinating, where they produced animals that completely lack rods and cones, and these were called rodless, coneless animals. And these animals were image blind, so they cannot see images. However, when they put them under light-dark conditions, you could see they photo-entrained similar to wild-type animals. So this was shocking that the results were published in Science, even though they didn't figure out which photoreceptor is responsible for that. So to make a long story short, it was really the pioneering work of Ignacio Provencio and David Burson, and then in collaboration with us, when I was a postdoc at Johns Hopkins University, that we now know that there is a subset of retinal ganglion cells that are intrinsically photosensitive, or IP, intrinsically photosensitive. So we call them IPRGCs, because they express an opsin similar to the rhodopsin and conopsins that can absorb the photon energy. And this opsin is called melanopsin because it was first discovered in the photosensitive skin melanophores of the frog. So then what happens if you remove melanopsin from IPRGCs? When you do that, you actually lose the intrinsic photosensitivity of these RGCs, but the animals still photo-entrain. So we wondered if there are other yet to be discovered photoreceptors in the retina. So to answer this question, we produced triple knockout animals where we knocked out rods, cones, and IPRGC's photosensitive functions. And when we did that in these triple knockout animals, now you have an animal that, although have a beautiful circadian clock, you could see it's beautifully running on its internal time, it cannot adjust to the light-dark environment. So this told us beyond reasonable doubt that there are only 3 photoreceptors in the retina that are responsible for circadian photoentrainment: the rod and cone system and these intrinsically photosensitive retinal ganglion cells. And that these intrinsically photosensitive ganglion cells are very interesting sensory cells because they not only could respond intrinsically through the melanopsin protein, but they seem to be able to also incorporate rod and cone input in the retinal circuit similar to the conventional ganglion cells. So since these IPRGCs are photoreceptive and ganglion cells, we wanted to trace where they project in the brain. And to make this possible, we actually labeled the cells and their axons with a blue dye. And what you could see here, all these dots are the IPRGCs, and these lines are their axons that leave the optic disc would be located here and go to the brain. And when we looked in the brain, we were really stunned at the specificity of innervation to the suprachiasmatic nucleus, the central of the circadian pacemaker that I told you before. So this is the optic nerve, this is where the eye would be located, and these two nuclei here, magnified here, are the SCN, the location for the central pacemaker for circadian rhythm. They also project to the LGN complex that is important for vision, but they avoid the dorsal part of the LGN, and they project specifically to a region that has been implicated in circadian photoentrainment, known as the IGL, which I'm going to tell you about later in my talk. In addition, they are not only circadian photoreceptors because they also project to the olivary-pretectal nucleus, which is important for pupil constriction. And when we looked at all the brain regions that these IPRGCs project to, we were actually stunned at the wide input to the many different brain regions. So in addition to circadian photoentrainment and pupil centers that I told you about before, they also project to sleep and alertness centers, as well as to mood regulating centers, and I'll be telling you a lot about a very exciting area that we discovered we call the perihabenular nucleus in this talk. And just to make the story full, actually all the studies I've showed you till now were concerning only a single population, which we now call the M1. However, work from these three amazing women in the lab have discovered that there are at least 5 different subtypes, maybe 6, that can be differentiated based on very specific morphological and electrophysiological properties. And in fact, Tiffany Schmidt, who has her own lab now at Northwestern, have really studied these very interesting M4 cells, which are important for image-forming functions. And I will encourage you to check her lab work to see their function in image formation. But before we even knew about the diversity of IPRGC, Louis Goulaire, who's now at UVA, he's a faculty at UVA, and Jen Ecker, who was in the lab, and you've seen her in the previous slide, decided to ask a very simple question. What would happen if you not only remove the melanopsin pigment from IPRGCs, but you kill the cells completely? And to do that, they expressed the attenuated diphtheria toxin A subunit under the melanopsin promoter. And the diphtheria toxin A subunit is a translation inhibitor that requires the beta subunit to infect the cells. So if you genetically express only the A subunit, then you specifically kill the cells that only express this gene. So this way you can specifically kill IPRGCs without killing the conventional ganglion cells. And in fact, this experiment worked really well. So this is where the IPRGCs are with the tau-like-Z animals. And now if you add the ADT alleles, we could see you could kill all these IPRGCs. Some of them escape, but you kill between 85 to 95% of IPRGCs. And if you look at the brain targets of these cells, you could see that the brain targets, although intact, their innervation from IPRGCs is completely ablated, or nearly completely ablated. Importantly, and I'm not going to show you the data, the innervation of the other non-M1 IPRGCs and the conventional RGCs are intact. What is so cool about us using ADTA animals is that this ADTA toxin only ablated the M1 IPRGCs, but not the non-M1 IPRGCs. In addition, this toxin, because it's the attenuated form, only killed M1 IPRGCs at adult stages. So this did not cause any developmental changes that could have affected vision or the function of the retina. So we were very lucky that we somehow developed a conditional line where we specifically eliminate one population of IPRGCs without affecting the development of the system. So then we asked the question, can these animals see? And in fact, these animals are completely normal in vision. We did very many different behaviors to show that they have normal image forming functions. But when we looked at their non-image forming functions, we were stunned to find that they have major deficit in the pupil constriction. So, for example, you need nearly sunlight level to get a constriction that is even much weaker than what you observe under moonlight level in the wild-type mice. And what was really stunned me when we did these experiments is that these animals, although are image capable, they are unable to adjust to the light-dark cycle, similar to the triple knockout animal that I showed you before. So this allowed us to make a major conclusion that there is a evolutionary separation between image-forming functions and non-image-forming functions, specifically at the level of the M1 IPRGCs. So now that we have all these tools to understand how light can affect circadian rhythm, and we have all these molecular tools to disrupt a certain pathway but not other, we wanted to revisit a very interesting phenomena that has been known in the circadian and the sleep field for many times. Namely, that irregular light schedule affect mood and cognition in a human. And these irregular light schedule include shift work, traveling across different time zones. Is something I would have done if I had come to India to present this seminar, which I would be much happy to be in India, but now I don't have at least the jet lag. And also the shorter day length of the winter month, which we start experiencing now in the Northeast in the United States as well. And what is common about these different irregular light environment is that they always have depression and learning difficulties. And the general idea in the field was that irregular light cycles affect circadian rhythm, which then affects sleep, which then affect mood, and then cause learning difficulties. But we thought maybe we're missing something because of a couple of things. One is that this environment doesn't have an irregular light. It's actually still have a 24-hour light-dark cycle. It's just the day length is shorter. So why would you have a problem in circadian rhythm if the day is still 24 hours? And the other thing is, I showed you before that IPRGCs project to brain regions that do affect mood directly. So we wondered whether light can have direct effect on mood and learning and memory, independent of circadian and sleep problems. To do this, though, we had to come up with a different cycle than the one I showed you before. So I showed you 12 hours of light, 12 hours of dark, or T24 cycle. But to be able to answer this very simple test, we had to produce a light environment where we're able to present light at all phases of the activity sleep cycle, but somehow not cause sleep deprivation or circadian rhythmicity. And I was lucky enough to have really two amazing women, Cara Altimus and Tara Legate. Cara works for the Milken Institute, and Tara now has her own lab at UMBC. And they came up with this cycle where it's 3.5 hours of light, 3.5 hours of dark, repeated for 2 weeks. So this cycle has been used in the circadian rhythm field, but people always use it with animals that have wheels. But they decided to use it for 2 weeks and without providing the animals with wheels. So we call it the T7 cycle. And that was their point of brilliance, because when we did that without the presence of wheels, the total sleep amount in the T7 cycle was not changed. The architecture of sleep, the amount of REM and non-REM sleep, was also not changed. Even the circadian clock was still intact. So if you look at the circadian clock, it's not arrhythmic. It's longer than 24 hours. It's not as beautiful as the T24 hours animals, but clearly it's still functional and you still have circadian rhythm. And what is really exciting to me when they showed me the data is when you overlay the light-dark cycle on the activity cycle, now you could give light at all phases of the circadian cycle without disrupting sleep, without causing circadian arrhythmicity. So now we could answer this very simple question with whether light has a direct effect on mood and learning and memory by maintaining animals either under the T7 cycle or under the T24 cycle. So the first test we did is a very well-known test in the depression field, which is the forced swim test, where you put a mouse in a beaker with no ability to escape, and the mice that have higher depression-like behavior, they will give up more, so they'll spend more time immobile. And what you could see clearly is that the animals that are maintained under the T7 cycle have more immobility, indicative of higher depression-like behavior. Another test that is much more accepted and much more humane to the mice, to be honest, is to provide the mice with 2 bottles of water, one spiked with sucrose. And again, mice that have depression-like behavior, they drink much less time from the sucrose water compared to the mice that have no depression-like behavior. So for example, you could see here that mice under T24 cycle drink 70% of the time from the sucrose spiked water, where mice under the T7 cycle, although still drink more from this sucrose water, much less than that of T24. Again, indicative of the loss of pleasure-seeking behavior, which is associated with depression-like behaviors. So to study learning and memory, we use many behaviors. I'm going to show you only one, which is a hippocampal dependent function. It's called the novel object recognition test. In this test, you put a mouse in a cage with two similar objects. Then you take them out and you put them back in the same cage, but now you provide them with a novel object. And believe it or not, mice are very inquisitive like us. When they see something new, they want to investigate. So what you could see here, mice under T24 cycle, because they remember seeing this object, they know this is new, they spend 70% or 7 minutes of the 10 minutes you measure them investigating this object compared to this object. But if mice have problem in learning and memory, then they don't show this attach or this higher investigation for this object, and they actually spend equal amount with both objects, indicative that they may have learning and memory problem. And as I told you, this requires a region in the brain known as the hippocampus, and the hippocampus usually is, when you stimulate it in vitro, produces long-term potentiation, which you see here in T24. And what you see, although you use the same potentiating electrodes in the hippocampi isolated from the T7 mice, you get much less induction of long-term potentiation in these brain, indicating that the learning deficit happened at the level of the hippocampus. So now that we showed that irregular light can directly affect mood and learning, independent of circadian and sleep problems, we wanted to ask, does this require the conscious image forming pathways, or does it require the non-image subconscious pathways? So to do that, we used the animal that I showed you before, where we eliminated only the M1 IPRGCs. And when we put these specific animals under T7 or T24, now because we don't have the M1 IPRGCs, the light has not been disruptive for mood behaviors. It has not been disruptive for learning and memory, and the LTP was superimposable between the T24 and the T7 cycle. So remarkably, what we found is that under the T7 cycle, just changing the light environment on animals without changing sleep and circadian rhythm causes depression-like behavior in mice. And this depression-like behavior requires the M1 IPRGCs, because if you eliminate them, you don't observe these depression-like behavior under the T7 cycle. And I didn't show you the data, and if you give antidepressant, you could also reverse these negative effects of the light-dark cycle. So now we changed the model that people had in the field, and we said, yes, light can affect, it's possible, we're not going to argue if it's yes or no, light could possibly affect mood and learning through circadian phase and sleep, but we think there is an underappreciated pathway where light through M1 IPRGCs can directly affect mood and learning and memory. And what's remarkable for me is that the image forming pathways, or the conscious visual perception, does not seem to participate in these negative aspects of light on behavior. So then we wanted to know which brain region is responsible for these functions. And Diego Fernandez in the lab took this project to really beautiful conclusion, as you will see now. And the first thing we wanted to see whether the SCN is required. We checked up the SCN because it's the central pacemaker, and because it has a beautiful circadian rhythm. Here's of a clock gene per 2, which has an incredible change across the day-night cycle with a huge induction in the evening. And to be able to study specifically the SCN, we had to have a way to only keep the SCN innervated while killing all the other IPRGCs. And luckily, we have produced this in 2011, and I'll show you how, where we found that IPRGCs can be molecularly subdivided into two major molecular rests, the transcription factor brain 3B, and that constitute the ones that project to pupil center, as well as all non-M1 IPRGCs, and one that doesn't express the transcription factor brain 3B, and these specifically project to the SCN. And this really was the work of Alan Chen and Melissa Semen. Both have their own labs at National Taiwan University and Oakwood College, where they mated the melanopsin Cree line with a brain 3B LOX-P stop DTA line. So what happens that cells that express both of these promoters will end up removing the stop codon and allowing the DTA to be expressed in cells that express both brain 3B and the melanopsin gene. And when Alan and Melissa did that, it was quite remarkable to see that the OPN regions is completely denervated, but the SCN region still maintains nearly normal innervation pattern. And consistent with this innervation pattern, they observed major deficit in pupil constriction, similar to the one we observed in animals where we killed all M1 IPRGCs. But they had normal circadian photoentrainment. So you could see that these animals that have the brain 3B positive IPRGC ablated, beautifully photoentrained, have a normal circadian clock similar to the wild type animals. And what's really, as I mentioned before, what was really incredible about this animal is that the conventional RGCs are there, the M1 that project to the SCN are there, but all other IPRGCs have been ablated. And so then Diego wanted to look at all the RGCs. They don't want to look at the whole RGCs that project to the brain. And what you could see here when you look at the SCN is beautifully innervated. The image forming centers are still innervated, but all the regions that are innervated by IPRGCs that are distinct from the SCNs are beautifully denervated, as you could see here. And since these animals still have the SCN innervation, the same observation we've seen in the wild type animals under T7 we see in these animals, where this circadian clock is lengthened but it's still functional. So now we could ask the question, what would happen to mood and learning and memory when you put these animals that lack the brain 3B positive IPRGCs under the T7 cycle? And I'm showing you the learning and memory for a reason first. You'll see why. When we put these animals under a T7 cycle, here's the controls. You could see, just like we published in 2012, that there is a problem in learning and memory in these animals. And these SCN-only innervated animals also have problem in learning and memory. And this is also reflected in a weaker LTP induction in these animals compared to the T24 cycle. However, when we looked at depression-like behavior, we were stunned, because although the controls show the previous results that I showed you before in the force swim test, these animals show no depression-like behavior despite having problems in learning and memory, both in the force swim test and the sucrose preference test. So this really excited us, because this showed, first of all, that the effect of light on mood and learning are dissociable, number one. And number two, the SCN is not involved. So if the SCN is not involved, which other region could be involved? So one area we looked at is the perihabenular nucleus. And the reason Diego picked this area is because it has this beautiful, but the clock is completely disrupted in this region when you put the animals in T7 cycle, indicative that it's highly light sensitive and that light can affect this area independent of time of day. And this is completely different than what happens to the SCN in T7 cycle. The SCN in T7 cycle still has a beautiful circadian clock, because I told you that the clock is completely functional under the T7 cycle, but only this PER2 levels in the perihabenula is affected. So then we wonder whether this perihabenula is the reason that light can induce effect on mood. So to answer this question, again, Diego came up with a very cool idea, where he noticed that this perihabenular region is very light responsive. And then he used a very interesting genetic trickery. So he knew it was light responsive by looking at CFOS induction, where he used the transgenic mouse line, where you express the conditional Cree under the CFOS promoter. So cells that respond to light will express this Cree, and then you inject the virus in this region, but this virus is a Cree-dependent DREAD. DREAD is the chemogenetic way that you could activate neurons with the addition of CNO. So what Diego did, he injected the virus, then he gave a light pulse plus tamoxifen to allow this conditional Cree line to come in and remove the stop codon, allowing the dread to be expressed. Gave 4 more weeks for the virus to be expressed, and then he gives CNO to these animals that will have only the expression in this region, but maintained under T24 cycle. So first of all, let me show you that the experiment worked beautifully. This virus also had M. cherry to label the cells, and what you could see, every cell that have a CFOS induction also have the M. cherry indicative that also has the dread expression. And remember, these animals were maintained under T24, but only this region will be activated continuously by the addition of the CNO to these animals for 2 weeks. And despite the fact that the animals were under T24, just activating these brain regions irregularly caused the same depression-like behavior that I showed you before in the forced swim test, in the sucrose preference test, and we added another test that measured depression, which is the tail suspension test. So what is this region? How come people missed it before? Well, first of all, it's a very interesting region at the dorsal thalamus. And when you look at where this region projects, again, this is work of Lorenzo Espri in the lab, you could see that it projects to the infralimbic cortex, a region very well known in regulating depression, not only in animal model of depression, but also in humans depression. And what was really remarkable is that in 2 synapses, the brain 3B positive IPRGCs project to this PHB region and then really fan out to 3 very important regions in the brain that are important in depression and reward systems such as the nucleus accumbens. So then, now that we know that this area is sufficient to induce depression-like behavior, we wanted to know, is it also necessary for the effect of light on depression? And so Diego used another animal where he has a tetanus toxin expressed under the Cree promoter. When you remove Fromagena promoter, and Cree will allow to remove stop to express this tetanus toxin. And he injected an AAV Cree virus in this region. And as you could see, he injected large amount of this virus because he has to block all these neurons with tetanus toxin, which prevent neuronal signaling. And now he put these animals under T7 cycle, but reminding you that now these population of neurons cannot signal to the 3 regions I showed you before. And now that despite the fact that these animals were maintained under the T7 cycle, they did not show any depression-like behavior. So this is really interesting because this region is very close to another area that is important for depression, which is the lateral habenula. But lateral habenula is important for depression for stressful conditions that doesn't require light, while this region seems to be specifically required for modulating mood for light responses. So just to quickly summarize to you then, we know that just putting the animals under the T7 cycle, you could induce depression-like behavior. Remarkably, even if you put them under this cycle, but now you inactivate this PHB region, you prevent the depression-like effect. But even if you put them under T24 and now you activate this region, you induce depression-like behavior. So now we discovered a completely new brain region where light, ambient light from the environment through this region can modulate very important regions that are known to be involved in reward and depression. And we can actually modify our model even further to say that even the direct pathway that is required for light effect on mood and learning and memory require 2 distinct circuits. And it's really very interesting how science works, in that the SCN, which usually has the clock, still have a functional clock at T7 but have a learning deficit, while the PHB, which is not a major part of the circadian clock that induces mood-like behavior, seems to have a disrupted clock under the T7 cycle. And in fact, a different population of IPRGCs, the brain 3B negative versus the brain 3B positive, the brain 3B negative versus the brain 3B positive, can regulate learning and mood independent of each other. One last thing I would like to tell you about this area is that it's found in this region that is very exciting, because it's this very small region close to the epithalamus where the melatonin is released for sleep. It's beside the medial habenula, which is important for addiction and nicotine addiction. The lateral habenula, which I told you is important for stress, and PVT, another very important stress regulating area. So it seems that these brain regions have been evolutionarily involved in many aspects that detect environmental stimuli and affects animal behavior to enhance mood or change mood depending on seasonality. So what I have shown you thus far is that there's this very simple light detecting system that seems to control many aspects of human physiology, including circadian photoentrainment, pupil constriction, but also mood and cognition. And these functions are dependent on specific brain regions. And interestingly, we recently found that light can directly affect body temperature and sleep, again independent of the circadian clock. Although the circadian clock can control body temperature and sleep, light effect can directly project to these areas and control sleep. And I wish I have the time to tell you about some of very exciting data to show that part of the VLPO is responsible for the direct light effect on sleep. But in the last maybe 10 minutes or so, I would like to tell you a very interesting story about the continuation of the importance of light for many aspects of animal behavior. And this work has been recently published, where we found, I'll give you the summary and then show you the data, that the anticipation of food, if you give animals food at a very specific time every day, the anticipation of food is detected by oscillators that we don't know yet where they are located, and through the IGL and release of the neurotransmitter MPY inhibiting the SCN, we think that this allows the animal to figure out when the food is going to be available, and that the IPRGCs themselves from the retina during development are important for setting up this circuit. I know I said a lot, but I'm going to show you the data to show you how exciting this new data is. And you're going to recognize that this work has been done with Diego, but also a very important person in the lab, Ruchi Komal, who comes from the University of Lucknow. So I'm very proud that she joined my lab, and I'm happy with her contributions. So this is what they did. It's a very interesting experiment where they gave animals first. So first of all, they kept all animals in constant darkness, so they don't have any input from light. And then they gave them food the whole time, or ad libitum, or they restricted the food for 7 hours during the daily cycle, because this is constant darkness. And if animals know the presence of food, they will start anticipating the food 3 hours before the food coming in, and this is called the anticipatory food behavior. So let's see what happens when you record feeding and locomotor activity in this paradigm from wild-type animals. What you could see is that even wild-type animals under ad libitum conditions, they restrict their feeding to a specific time of the day-night cycle, especially when they are mostly awake and active. And when you restrict their feeding to the 7-hour time, they only restrict their feeding to that time you're giving them food. But what's really remarkable, what you could see, they start anticipating this food, and they start having a higher activity just before you give them the food pellets. And when you combine the feeding and the activity together, what you could see is that the feeding and activity anticipation have a very beautiful correlation with each other. So the first question we ask, do IPRGCs play a role in the development of the system? And what we use animals different from the ADT animals that I showed you, these animals that express the full strength diphtheria toxin A subunit. And what happens in these animals that IPRGCs are killed at developmental stages. So now you don't only kill IPRGCs in adult, you kill them developmentally. And when you do that, what you notice is that the feeding of the animals looks the same as the wild-type animals, but now the anticipation of the feeding response is completely gone. Even when the circadian clock is actually trying to interact with the feeding time, there doesn't seem to be a feeding anticipation to the presence of food. This was shocking to us because when we first did these experiments, we expected that if you lose the light input, you will have a higher sensitivity to non-photic stimulation, which is feeding is a form of a non-photic stimulus. We thought, similar to the case of blindness, for example, it is known that patients who are blind, they usually have higher level of auditory and somatosensation. But in this situation, it was actually the opposite of our expectation, is that when you lose light reception for the circadian clock, you also seem to lose the non-photic input as quantified here. What you could see is that the anticipation activity in the OPNDK animals is much weaker, both in this way of showing it and this way, than the control animals. So then we ask the question, why does this happen? And what we notice very interestingly is that the MPY input to the SCN is highly reduced. So what is something that is really interesting in the circadian field is that the SCN receives input from the retina, but also receives input from the IGL. And this IGL input to the SCN is mostly through the neurotransmitter MPY. And what you could see in the control animals is that there is retinal innervation, there is MPY innervation, and they overlay pretty nicely with each other. But now if you kill IPRGCs in early development, as you would expect, you don't get innervation of the SCN because it's mostly innervated by the IPRGCs. But we were also surprised that you lost the innervation from the MPY neurotransmitter. And consistent with the loss of innervation, what we notice is that the IGL is very responsive to the time-restricted feeding paradigm. So when you give the animals this feeding at specific time of the day, you get a beautiful activity in the IGL of wild-type animals just before the presence of the food. This activity or this sensing of the IGL of the food presence is not as robust when you kill the IPRGCs, indicative that the ability of the animals to detect the time-restricted feeding paradigm has been completely reduced in the absence of IPRGC innervation developmentally. The question is, does this happen in adult animals? And so we use now the melanopsin attenuated diphtheria toxin that I showed you before, where it only kills IPRGCs in adult. And in these animals, as you could see, the animal beautifully anticipates, similar to wild-type animals. So there is a very specific dependent time window by which IPRGCs are responsible for setting up this time-restricted feeding behavior. And we did these experiments to find when does this happen. So when we removed the eyes from mice at P0 or at P40, we got a major reduction in the anticipation of food. But if you remove them in the adult at P90, now you get normal anticipation. So it's remarkable that up to P40, up to 40 days after the mice are born, the melanopsin system or the IPRGCs and the retina are still required for setting up the best aspect of non-photic entrainment in the animal. And that to me is quite impressive because it tells you that the eye has been chosen for some reason to organize all these daily activities to both light-dependent behavior, which is not surprising because the eye is the light-sensitive organ, but also probably to behaviors that do not require light and are collectively known as non-photic behaviors. So finally, we wanted to see, does NPY play a role? And to do this, Diego got NPY Cree animals. And so what he did in these animals, he got the heterozygous animals, and if you inject the virus that doesn't have tetanus toxin, these are the control animals, you could see that the NPY Cree animals beautifully anticipate to the food time-restricted feeding paradigm that I showed you before. But now, if you inject in the NPY Cree animals that you could see here labeling the IGL, a virus that would allow them to express the green fluorescence protein and tetanus toxin, which prevent them from signaling to the SCN, now you could see very nicely that these animals, even in the adult, can have a problem in food anticipation. So this is why we came up with our model that actually what happens is that for time-restricted feeding, there is some oscillator somewhere that sends this information to the IGL. That's why you get beautiful CFOS induction in the IGL. And then the IGL inhibits the SCN, which usually inhibits the animal's ability to be running around at a different time of the day. So what happens when you have double inhibition, you now start anticipating the presence of food, and you know when food is going to appear. And remarkably, it is actually IPRGCs themselves that are required for setting up the system to function for non-photic entrainment pathway. So this also told us that similar to the mood and learning and memory, the systems are not very simple. They are actually quite robust and quite exquisite in a way where you need a system to set up the behavior where you not only think of feeding now as just hunger. So the hunger is important possibly through the arcuate, but there are other behaviors that requires the SCN for photo entrainment of feeding, but also that tells the animal when is food going to be available for them, and that the combination of all these behaviors are going to be very important for the survival of the animal, and tells us in a way that even though as animals we don't depend on photosynthesis for survival, light seems to be the central component. Light and the eyes and IPRGC seems to be the central component that are important for organizing our circadian rhythms, mood, learning and memory, and even the time of feeding. And with that, I would like to thank all the people who helped us do the work. Tiffany Schmidt in her own lab that allowed us to find that sleep and temperatures do not require the SCN. Tudor Badia, who actually we first worked with him on the Braintree bee animals, David Burson and Michelle Ferguson on the PHP region, and Alfredo Kirkwood for all the LTP recording that I showed you on when I was on the outside, all the funding from the extramural areas, especially the Packard Foundation, who first gave me the money to do the mood and learning and memory stuff, and now all the funding that is provided by the intramural program at the National Institutes of Health, and specifically the National Institute of Mental Health. And with that, I would love to take any questions if anybody has any. I could stop the presentation. I don't know if you see me or however you want to do it.
Samer, as usual, it was very interesting to hear you.
Thank you.
And it was more interesting because as it is a general myth that a person who is blind forever, so it's not like that. As you have very clearly mentioned that IPRGs play a major role. And so our pupils and the circadian system are totally different systems, go on differently. So
yeah, and just to add to this point, Professor Malik, I think something I failed to mention, that in fact people have noticed that there are two types of blind people. There are the blind people who will only have image blindness but have no problem sleep-wake cycle.
Right.
And there are blind people who have both image and circadian. And in fact, these blind patients have the same thing as I showed you in the triple knockout animals, where you could see that they have a cyclical sleep problems depending on their circadian clock. So it's very interesting that, as you mentioned, blindness can be image blind and circadian blind and so on and so forth. So this is something we need to think about these days. Absolutely.
Yeah. The questions, one of my colleagues is taking the questions. She'll just give me and then I'll ask you. Before that, I can ask you a quick questions, which I could understand. You have the answer, I'm sure. Like, why is it that IPRGs are sensitive for time-restricted feeding? Means, why do you think so? You have shown that. But
I think this is where I would love to get the insight of the zoology department. I think them really the vision and hearing, although they interact with each other, they are sensory modalities. They are very independent sensory modalities that do specific function. But I think for the circadian system and time restricted feeding, you somehow still have to organize everything as a system. So evolutionary, it doesn't make sense that non-photic information doesn't have information from the eye. So I think it makes more sense if the system, although independent, is organized into a central theme by the retina and the SCM. We were really shocked. We did not expect this data, but that's the best answer we have right now. That's a tough question. Yeah.
Now here are some questions from our students. So it's there in the chat box. There's a question: if the RPRGCs can cause photoreception, stabilize the circadian rhythm, and are also present on the outer periphery of the retina, why are rods and cones called primary photoreceptors and not the IPRGCs?
Okay. That's actually a great question. I think it's possibly so if I would say which photoreceptors possibly evolved earlier, I would say IPRGCs. And considering their major function for so many behaviors, clearly they have more important role in the physiology of the animal. But I think it's a matter of when they were discovered. So, rods and cones have been the leaders in the retina for a long time, and IPRGCs are new. But I take that point. I would say they are absolutely the primary photoreceptors for non-image forming functions. That's for sure. That's a great question.
So do you think the IPRGCs they are conserved, they are evolutionary conserved in different species, if we say birds, rats, or like mammals, birds, or reptiles, or other groups?
Yeah, I don't know if any vertebrate animal that we didn't find melanopsin in some form of ganglion cells. So it seems to be very conserved. One of my hypotheses, which I don't know how it's going to be tested, is that maybe ganglion cells were the first photoreceptors that would drive behaviors, and then rods and cones were added to the system. And once you have these beautifully evolved photoreceptors, they are more taken for vision, and IPRGCs have kept their function for non-image forming function. One thing for sure is that it's found in all vertebrates, so it clearly has a very important function in every vertebrate animal that we tested, mammal or non-mammal. Even the blind mole, for example, the blind moles who live underground, whose eyes degenerate, they still seem to have a lot of melanopsin expressing IPRGCs, and they circadianly photoentrain to light, even though their eye is under the skin. So that's really interesting. Yeah.
Student, what kind of difference we can observe in photoreceptors and behavior of animals living in extreme poles where light is for almost 6 months?
Yeah, that's a really good question. So one of the things that we have been talking about the lab a lot is that we always measure light. That's a really good question, by the way. Light dark in a way that you give light, you give darkness. It's a very square reaction. But I think another way the system can count photons, and because of the IPRGC sensitivity, they possibly can entrain even if the light is still there, as long as there is a variation in the light environment. So that's one of my predictions, that there may not be a clear light-dark cycle, the intensity of light could determine a day. Or they could be like some of the animals that live in these extremes, that their circadian clock goes completely out of whack in these months where the sun is always there, and they actually don't have the circadian clock. So they'd be functioning along the day-night cycle, or day-day cycle. So it's a very interesting question. I could only, wave my hand and try to answer it in a certain way.
Okay. There is another question from one of our colleagues. Are human circadian rhythms influenced by EMR?
What is EMR? I'm sorry, I'm not good in shorthand.
Gitanjali, are you here? Is it electronic something, EMR, or?
It was asked by a student on the
YouTube.
Yeah, it was asked by a student.
I think it was electromagnetic radiation. I think so.
Electromagnetic radiation, yeah.
Yeah.
Is what? Sorry.
It's electromagnetic radiation.
I don't. So it's really interesting. So cryptochromes, which are the photoreceptors in flies and sometimes in birds, I'm not so sure how much it does photo entrainment. Even in so for example in butterflies, I know cryptochrome play a major role in circadian photo entrainment, dependent oscillator of the SCN. So I don't have the answer to that.
So these were the questions. Now I hand over the baton to our head of the department, Professor Sudhir Kumar. Sir.
Thanks, Professor Samar. So, just to lighten it up. Actually, nobody was fit. Like,
I'm sorry, I can't hear you anymore.
hello.
Yes, now I can hear you. Uh-huh.
So the motto of this university is light and learning.
Yes.
So that is a good start or good beginning for this university. And now the animals or what is the role of light in learning and the non-photic and photic behavior. So I am thankful to you for spending your time for our students and the faculty. I would love to have you here for your video projects, and I'm also able to train our student that is who is working in your lab. Thank you again, and
Thank you so much. Thanks so much for the invitation and congratulations. And hopefully many more years to come. Success and more students and better future.
Thank you.
Everyone, see you later.
Good night.
Good night.
Anurati?
Yes, ma'am.
Anurati?
Yes, ma'am. Yes, ma'am.
Anurati?
Yes, ma'am.
You can put the...
Okay, ma'am.
So we'll all be back at 11:30.
11:30 Gitanjali or 11:20?
I think, yeah, 11:20 will be fine. We should just stay connected. It's just another 5 minutes.
Yeah.
10 minutes.
That's right. Sure. Okay.
Morning, sir.
Morning.
How are you, sir?
Good, good.
Yes, sir. So shall I after the proceedings?
One second.
Hi, please just let me
I
know when
can
you.
just share the screen.
Yeah. Anuriti?
Yes.
Does the screen come up?
Yeah, we're just checking it on YouTube as well, so sure. I think it should. Anuriti? I actually can't see your screen. I saw it for a moment, but yeah, now it's visible. Yeah.
Oh, I see. I think I have to be in that window for it to be visible.
Yes, exactly. The drawbacks of Google Meet.
Okay, fine. Then then we can proceed.
Yeah, just a second. Let me check with the student who's screening to be sure that she is there. Anuriti,
Yes, ma'am.
are we live on YouTube now?
Yes, ma'am.
Just ensure.
Yes, ma'am, we are.
And we can see your presentation, right?
Yes, ma'am.
Okay. Yeah, great. So then we'll start. It's a pleasure to have you. For all our viewers, I'll give an introduction to you about Professor Joshi. So Professor Amitabh Joshi is an Indian evolutionary biologist, geneticist and a professor at Jawaharlal Nehru Center for Advanced Scientific Research at Bengaluru. he heads the Evolutionary Biology Laboratory and is known widely for his studies in evolution, genetics, and population ecology. He and his students have been using laboratory populations of fruit flies to address various questions falling under life history, evolution, and population ecology and their interaction. He is a fellow of all the three major science academies of the university. He won the Shanti Swarup Bhatnagar Prize for Biological Sciences in 2009. The Young Leader in Science and Technology by Lakshmipat Singhania National Leadership Awards in 2010. Professor Harsharup Memorial Lecture Award of the Indian National Science Academy in 2017. He is the fellow and founding member of the Indian Society of Evolutionary Biologists 2018 and an executive council of the same. He and his fellow members of the Indian Society of Evolutionary Biologists are extremely passionate about spreading the word about and awareness about how important evolution is and central to our lives, which will be the theme of what he'll be talking about. A special mention that one of his papers in Biology Letters, which he co-authored along has traded off with other traits. Also, Professor Joshi is deeply interested in Urdu poetry, Persian poetry and folklore, older English poetry, Sufism. He balances his love for traditional Qawwali in Urdu, Persian, Braj Bhasha and Punjabi with his delight for hard rock, especially Bob Dylan, Leonard Cohen and Chris Christopherson. And he himself writes lovely Urdu poetry, his pen name being Nabras. So a multifaceted personality whom you will be hearing today about why evolution is central to both biology and our lives. The stage is all yours, Professor Joshi.
Thank you, Dr. Gitanjali, for a very, very kind introduction. It's wonderful to be here, especially as these talks are to celebrate the centenary of Lucknow University. I'm originally a native of UP. I grew up in Agra, not all that far from Lucknow. So today's talk is really a very fairly general talk about why evolution, rather than being a marginalized backwater of biology as is often thought in India particularly, it is actually central not just to biology as an academic discipline but also to our lives as a human society faced with multiple problems. So in the talk today, what I'd like to do is first of all sort of take up and bust 4 very common myths about evolutionary biology. And included in this is discussing very briefly why evolution is central to biology, in a little more detail why evolution is central to our lives. And finally, towards the end, I would like to talk to you a little bit about evolution in the Indian context, because this is not very well known, but Indian evolutionary biologists and ecologists over the decades have actually made fairly profound contributions to the growth of biological knowledge worldwide. So 4 very common myths about evolutionary biology. The first one which you hear all the time from people is, oh, evolution is just a theory. As if that implies that it's some kind of weird speculation with no proof or evidence supporting it. And I'll talk about this in a minute. A second thing I feel that many people keep thinking, and this often shows up in the media as well, is this feeling that many of Darwin's ideas about evolution have subsequently been proven to be wrong. this is also really just a myth. Third, evolutionary biology is no longer an active research field. I have actually had the experience about 15 years ago of giving a talk in the zoology department at one of the very well-known universities of the country. And 10 minutes into the talk, the chairperson of the department stood up and said, "Sir, anyway, Darwin already told survival of the fittest, so what is there to do research in evolution?" So this is also a myth. It's a very active research field, as I will hopefully convince you. And the fourth myth I want to bust is that evolutionary biology is of academic interest only. It doesn't have any applied significance. When we think of practical applications of biology to solve societal problems, we are perpetually thinking about vaccines or some kind of new device, some new drug, some new genetically modified crop, etc. We rarely think of evolution as having applied significance. And yet, especially right now as we are in the middle of a global pandemic, the lacuna that we have in terms of not training people in evolutionary biology and ecology is showing up in India very badly in the paucity of epidemiologists that we have. Because epidemiology is really evolution and ecology applied to human diseases. So there is a centrality, and this I will talk about in some detail later on in the talk. So to first come to the point that evolution is just a theory, this actually arises because the common language usage of the word theory is somewhat equivalent to a wild speculation. But in science, theory is diametrically opposite to a speculation. The term theory really refers to scientific theories, a well-confirmed type of explanation of nature, made in a way consistent with scientific method and fulfilling the criteria required by modern science. Such theories are described in such a way that any scientist in the field is in a position to understand and either provide empirical support or to verify, or empirically contradict or falsify the theory. In fact, in science, when we say something is a theory, we are calling it the most reliable, rigorous and comprehensive form of scientific knowledge. So yes, evolution is a theory, but if we want to claim that it's just a theory, then yes, it is just a theory, like gravitation is just a theory, electromagnetism is just a theory, the theory of projectile trajectories is just a theory. In fact, our whole modern world is based upon applications of all these just a theories. So theory in science doesn't mean a wild hypothesis, it means something that has tremendous support experimentally or observationally. It also means something that meshes well with pre-existing levels of understanding. Now, of course, we all know that evolutionary biology in the modern sense began with Darwin. Most of us are familiar with the book on the left of your screen, The Origin of Species. But actually, Darwin's views on evolutionary biology are really spread out across 4 books. The Origin of Species, 2 volumes entitled The Variation of Animals and Plants Under Domestication, and finally, The Descent of Man and Selection in Relation to Sex. Now, the central insight that Darwin had was actually very simple. And this was that individuals that are better suited to make a living, to survive and reproduce in a given ecological situation will tend to have offspring that resemble them, and they will tend to have more offspring than other individuals that are less well suited to make a living and reproduce in those ecological scenarios. Therefore, over generations, those traits that made these individuals better suited will become more common in the population, eventually leading to substantial changes in the makeup of the population, potentially leading to the formation of new species. Now, this is a very, very simple idea, but it has tremendous implications. And in several of the slides that follow, what I will try to show is how much work built up on this original basic central insight of Darwin to form. I am, as Dr. Gitanjali mentioned, an experimental evolutionist. So I'm very happy to share with you that the first ever experimental evolution study was actually done within about 20 odd years of Darwin's Origin of Species being published in the 1880s. And William Dallinger, who you see on the left, cultured unicellular protists in an incubator, which you see at the bottom right. And initially, this species was a cold temperature adapted species. They showed major stress at about 24 degrees and died by about 35 degrees. What Dallinger did was to culture them and gradually increase the temperature of rearing from 16 degrees centigrade to 70 degrees centigrade over several years. And after a few years, the species could comfortably grow at 70 degrees, which used to earlier die at about 35 degrees, but it lost the ability to grow at 60 degrees as it adapted to high temperature. Dallinger regarded this as an experimental verification of adaptive evolution as proposed by Darwin, although it was not really verification, as it was an illustration of a process that would be consistent with Darwin's theory. It's not really a verification of Darwin's theory. This is also interestingly the first documented example of a trade-off between Darwinian fitness in different environments, in this case the ability to survive at low temperature versus the ability to survive at high temperature. Again, in the late 1800s, shortly after Darwin's book, one of the outcomes that happened was that field naturalists, what today we would call field ecologists or wildlife biologists, tried to interpret what they were seeing in their expeditions in the natural world in terms of Darwin's theory of natural selection. And two of the most classic examples of these Darwinian interpretations of patterns of variation observed in the real world are the so-called Batesian mimicry and Mullerian mimicry, which were worked out to explain why different species have some benefits in terms of protection from predators. And this began from merely being a descriptive kind of work to trying to unearth evidence of Darwinian mechanisms actually playing out in nature. But once again, in the initial phase, these explanations were not so much of natural selection. They were accounts of. This slide introduces you to Ernst Haeckel, a German biologist, a little bit younger than Darwin, who was perhaps one of the most influential biologists of the 19th century along with Darwin and Mendel. He coined the terms ontogeny, phylogeny and ecology. He interpreted systematics in a Darwinian framework for the first time in a big way. He elaborated on older ideas of why embryonic stages of vertebrates are more similar to each other than their later stages are, gave it an evolutionary interpretation. And what is not so well known about Haeckel is he was a great science communicator, as we call them today. He gave public lectures, he wrote books for the general reading public, and he was also a great poet in his native language, German, and he was a very good artist, as some of the pictures on the left show. These are his own drawings. In fact, a collected book of his drawings and illustrations of species was released as the Kunstformen der Natur, or the artworks of nature. And Haeckel was also the first person to appreciate that a Darwinian view of the living world would eventually also begin to have implications for how to understand human societies, human social behavior, and so on. In fact, he also had developed the idea of sexual selection before Darwin, and Darwin acknowledges this in the preface to his book on the descent of man. And Darwin actually says that had I known that Professor Haeckel had already written about these ideas in his German book, I would not have bothered, because he has already covered the ground so well. I now come to two people, Fleming Jenkin and Francis Galton. Galton was actually a cousin of Charles Darwin. Both Jenkin and Galton present an interesting example of how in science one can often be wrong and yet contribute greatly to the progression of scientific understanding. Both Jenkin and Galton thought that natural selection would not work in the face of heredity. Both Jenkin and Galton thought that heredity would act as a homogenizing force which would tend to bring populations every generation closer back to the mean phenotype. And therefore, if selection were, in a sense, tending to alter the population mean phenotype, heredity would tend to bring it back and therefore cancel out the action of selection. However, in the process of being wrong, they came up with many ideas. For example, Jenkin laid down in great detail exactly what information about heredity would be required to evaluate the role of selection in evolutionary change. And Galton, while he experimentally disproved Darwin's theory of heredity, which was via pangenesis and gametes budding off from every organ and then contributing to hereditary transmission, he developed the statistical approach to heredity and selection. And his statistical approach became the foundation of what was called biometry and underpins a lot of evolutionary theory even today. Galton also underscored the importance of Darwin's realization that heredity could be thought of as the degree of similarity of phenotypes or traits between offspring and parents, regardless of what the mechanism driving such similarity was. This was actually one of Darwin's great insights, which is not that well known. Darwin also changed the way we think about heredity. And he emphasized that for understanding evolution, what we really need to understand is how parents and offspring are similar. Not the mechanism by which they are similar, but the degree to which they are similar. Following up on Galton's work, there is some remarkable work done collaboratively by Raphael Weldon and Carl Pearson around the turn of the 19th to 20th centuries. Weldon, in fact, provided the first detailed example in which he could capture natural selection happening in the wild and did it in a manner embodying such exemplary rigor that even today his approach can hardly be improved upon. Basically, he showed that there was a shift in population mean phenotype over generations. He was able to link that shift to a change in the environment and then experimentally show that by providing that changed environment to crabs in an aquarium, you could mimic the size-dependent mortality pattern that he assumed had caused a shift in the natural population. This is actually, and this study was done around 1902 or so. This is the first solid verification of the entire mechanism of natural selection, and that too on a natural population. Absolutely amazing work. And as I said, in its rigor and its conceptual clarity, this is how we still demonstrate natural selection today. We have not improved upon Weldon's idea. Pearson, in his collaboration with Weldon actually developed many statistical concepts and theories that later became very important to the development of quantitative genetics in the hands of Fisher. In 1900, we are all familiar with the rediscovery of Mendel's work by von Schermack, Karl Lorenz, and Julie Breeze. And after the rediscovery of Mendel's work, once the putative mechanism of heredity became known, it became important to reconcile the Darwinian view of adaptive evolution through natural selection acting on small variations, small phenotypic variations, continuously varying traits, with this discrete Mendelian mechanism of heredity. And this was largely achieved through what is called the Neo-Darwinian synthesis over a couple of decades, through the remarkable work of people like Ronald Fisher, J.B.S. Haldane, and Sybil Wright. They really Fisher showed that Mendel's laws are compatible with the statistical understanding of heredity and with adaptive evolution through natural selection acting on small continuous variations. One fact that is actually not very well known even to students of genetics is that Mendel himself had appreciated and mentioned in his papers that if you imagine a trait controlled by a very large number of Mendelian genes, then that trait will have a continuous phenotypic distribution. Very few people are aware that Mendel himself had actually pointed this out. On the bottom left of your slide, you have two people, Theodosius Dobzhansky. Incidentally, I am, academically speaking, a great grandson of Theodosius Dobzhansky, and E.B. Ford, who respectively in America and in England started empirical programs of field biology and laboratory experimentation to test the predictions coming out of the population genetics theories of people like Fisher, Wright, and Haldane. And these programs eventually blossomed into major schools of experimental ecology and evolution, both deploying laboratory experiments as well as field studies which were observational, and also field studies that were manipulative experimental in nature. Subsequently, Fisher, Zahavi, and others extended Darwin's idea of sexual selection to understanding how selection through mating preferences rather than survival shapes a lot of how the living world looks. This theory eventually grew to explain intersexual conflict at the genetic level, which arises because the evolutionary interests of males and females are often quite different, because female reproductive output is primarily limited by access to resources, whereas male reproductive output is primarily, in most species, limited by access to females. And this creates a difference in what kind of traits are optimal for maximizing fitness in males and females, and this drives both interlocus genetic conflict between the sexes, which is sort of an arms race between males and females, and intralocus conflict, which arises because for the same trait, the optimal phenotype might differ between the sexes. Interlocus conflict, incidentally, can be a huge driver of speciation. The first experimental evidence of this has also come from an Indian lab in Aysar Mohali from Professor N.K. Prasad's group, and I'll talk a little bit more about that later. And finally, in the 60s to 70s, there were expansions of Darwinian thinking once the basic fundamentals were worked out. William Hamilton elaborated on Darwin's and Fisher's insight and formalized theories both of kin selection to explain the evolution of altruism and about the evolution of aging. Extremely profound work in 3 papers published in the mid-1960s. G.C. Williams in America elaborated on the gene's eye view of evolution and formalized the role of selection. Bob Trivers, one of the most interesting evolutionary thinkers of the second half of the 20th century, did a Darwinian analysis of human behavior, including absolutely path-breaking work on parent-offspring conflict, reciprocal altruism, self-deceit, and the evolution of human social and marital systems. Moto Kimura, who studied in Japan and then came to the US with a desire to work with civil rights, ended up working with James Crow, developed the entire theory of molecular population genetics of how DNA sequences evolve with or without natural selection. This work of his, particularly on the evolution of selectively neutral DNA sequences under the combined effect of drift and mutation, lays the foundation for almost all the ways in which we use DNA sequence data to construct phylogenies today. And finally, George Price, who was a maverick not trained in evolutionary biology or even in biology, actually came up with the most general formulation of natural selection that can work even with arbitrary heredity. In fact, it can be shown that Fisher's fundamental theorem of selection, which came about in the 1920s, is actually a special case of the Price equation under the assumption of Mendelian heredity. George Price's equation of natural selection works for any arbitrary form of heredity that you might come up with. And it again goes all the way back to the insight of Darwin and Galton that what really matters is the phenotypic similarity between parents and offspring, not the precise mechanism of heredity by which it came about. Excuse me. Have Darwin's ideas been proved wrong? The answer is absolutely not. I list here many of Darwin's insights. Species can give rise to other species. Descent with modification absolutely still stands. Natural selection shapes adaptation, still very much there. Sexual selection can explain extreme ornamentation and sexual behavior of various kinds, very much around. Kin selection can explain the evolution of altruism. Again, often people don't appreciate this, but long before Hamilton and even Fisher, Darwin, without using the term kin selection, had actually come up with the explanation that often a selective advantage could be accrued to an individual.
अरे the hell, अनुरीत Saxena, the hell.
By the wayside as we have progressed, is Darwin's insistence that evolution is always slow and gradual. And this was actually a philosophically inspired position rather than a biologically inspired one. Darwin was a great believer in the philosophical notion of gradualism, propagated first in the context of geology by Charles Lyell. And he insisted that evolution had to be a slow and gradual process. Today we realize that evolution, even in the wild, can often be very fast. It doesn't have to be slow and gradual. Sometimes it is slow, sometimes it can be very fast. But every other insight or prediction of Darwin's has been borne out by the following 170 years or so. Evolution is central to biology. I just want to say a couple of things here. There are some quotations here. You can obviously see them. The main point I want to stress here is what is mentioned right at the bottom of the slide. See, evolution is not a branch or a sub-discipline of biology the way morphology is, or the way biochemistry is, or anatomy is, or molecular biology is. Evolution is a logical framework within which we view all biological facts. It is a perspective. It's a pair of spectacles which you put on and then look at biology through them. Basically, biology has a certain logical structure and coherence to it as a discipline because of evolution. Biology without evolution would be somewhat like chemistry without the periodic table. It would be a huge collection of facts with no logical structure in which to place those facts in a pattern that gives rise to some understanding. It is evolution that converts biological knowledge or facts or information into an understanding and a logical structure. And as Peter Medawar said very eloquently, and Peter Medawar, mind you, was not an evolutionary biologist, so he cannot be accused of biasly favoring his own discipline. He was an immunologist who did Nobel Award-winning work on the major histocompatibility locus or loci. And Peter Medawar says, for a biologist, the alternative to thinking in evolutionary terms is not to think at all. That's a very strong statement. One more point I would also like to mention, especially because there are hopefully lots of students in the audience, is that in some ways there are actually two very distinct kinds of biologies that ask two very different kinds of questions. There is one set of questions that biology asks, which is on the top of the slide, and these are questions of the kind that seek to understand how is it that living organisms are living? What are the processes at the cellular or molecular level of life form? The mechanics of life, if you like. This is one set of questions, but this is just one part of biology. The second set of questions in biology is that when we look at the living world, we see that life forms come in very diverse varieties. we see that this diversity is not just random, but it is organized in a nested hierarchical pattern, which we call relatedness. So life forms can be grouped hierarchically into ever-inclusive categories based on relatedness or similarity, which is why we have a taxonomy at all. And finally, we realize that life forms have structures and functions that seem to be very suited to the ecological situations they live in. So there are also many questions about the living world that pertain to trying to understand the diversity of life forms, the relatedness of life forms, and the adaptedness of life forms. And this constitutes a huge part of biology, which unfortunately today is relegated into the background, and the entire focus in our curricula, our syllabuses, and our funding priorities is on this top part of biology, as if this alone were all of biology. And I would seriously suggest that just like we think of physics and chemistry as two sciences, but they both deal with the non-living world, reductionist or mechanistic biology, the top one here, and evolutionary biology, or the bottom one here, in many ways are as different from each other as physics and chemistry are from each other. Of course, just like physics and chemistry have some overlap and some fuzzy boundaries, the two kinds of biology also have some overlap and some fuzzy boundaries. But really, there are two very distinct kinds of biologies with very distinct methodologies and very distinct ways of thinking about the living world. Cellular and molecular biology embraces the approach of reductionism, whereas evolutionary biology, ecology, and the study of behavior embrace more the approach of holism, or a holistic approach to understanding. And indeed, many biologists would argue that just as William Butler Yeats, the great English poet, said that poetry is that which gets lost in the translation, many of us would argue that biology is that which gets lost in the reduction to precedent. Okay, let me now shift gears and come to why evolution is central to understanding and solving many pressing human and societal problems. What I have on the top row of this slide is just a small selection of relatively recent books to illustrate this. On the extreme left is a book entitled Why We Get Sick, The New Science of Darwinian Medicine. Especially for students out there, I would urge you to Google and look up Darwinian medicine or evolutionary medicine. You will be amazed at how much material you find. Basically, what we are now beginning to realize is that most of the biomedical problems that we face, whether due to pathological organisms causing infectious disease or whether due to the so-called lifestyle diseases like diabetes type 2 or hypertension and so on, or even cancer or even autoimmune diseases, all of them are better understood through a framework of evolutionary theory. Indeed, in cancer now, almost 30 years after evolutionary biologists started pointing this out, the oncology community has actually suddenly fully internalized the importance of an evolutionary approach to cancer, because what a cancer is an evolutionary process happening to that population of cancer cells within your body. Your body is like the environment in which a population of cancerous cells is growing and natural selection is happening among them, just as it happens among individuals living in the physical environment. So an evolutionary approach is actually crucial to understanding all kinds of biomedical conditions, and therefore it is also crucial to rethinking how we try to manage those diseased conditions. And this is a very rapidly growing field, unfortunately not much in India, but internationally this is now a very, very, very important field. The next book second from left, is called DNA Technology in Forensic Science. All of you are familiar with the use of so-called DNA fingerprints for forensic purposes to compare to DNA samples obtained from the crime scene and to establish paternity and so on in legal proceedings. What is not that well known is that the proper application of this technology to forensics requires a very, very thorough understanding of the theory of molecular population genetics, which is a core part of evolutionary theory. In the absence of that knowledge, applying in a very simplistic way DNA technology to forensic cases actually leads to major miscarriages of justice, and this is well documented around the world. The third book is The Moral Animal, Why We Are the Way We Are, the New Science of Evolutionary Psychology. This is again something worth Googling. There are many critiques of evolutionary psychology also, but those critiques are more about the often slightly unrigorous and cavalier manner in which some people have deployed evolutionary psychology explanations. The basic logic of evolutionary psychology is actually quite sound. And that is to point out that everything we think of as basic human nature, or you say you have an intuition about certain things, you have a gut feeling about certain things. All these basic intuitions that we have are also evolved characteristics. And in many ways, we can understand certain aspects of our psychological thinking, which of course then affects our behavior in an evolutionary perspective. And this is again a very interesting new extension of evolutionary biology to the human domain, first predicted by Ernst Haeckel in the 1860s. The next book here is Darwin's Spectre, Evolutionary Biology in the Modern World, which shows how evolution permeates modern biology, literally from agriculture to trying to understand why humans seem to believe in God. The next book here by two of my mentors, Lawrence Miller and Michael Woods, is called Does Aging Stop? And the brief answer is yes, it does. And the slightly more elaborate answer is that our understanding of aging has been completely transformed in the last 40 years, largely through the work of people like Michael Woods and Lawrence Miller. Drawing upon the formal theory created by Hamilton in the 1960s, they have brought about literally a revolution in how we understand aging. That revolution, unfortunately, has not yet percolated into the thinking of many biomedical researchers and many molecular biology researchers who still think that aging is all about telomeres or one of the genes that will somehow extend your lifespan, and the basic answer is it's not. And if anybody wants more details on this, I would strongly recommend you to look up Professor Michael Woods' website. It is called 55theses.com, 55theses.com. The last book here is Genetic Mapping and Marker Assisted Selection, very important technologies used in agriculture for the last many decades. Would not have been possible without evolutionary theory. In fact, if you think about it, agriculture or animal husbandry, animal breeding, is our attempt to divert the course of the evolution of certain crop species or domesticated species towards traits that we think are useful to us, which is basically directed evolution. And of course, plant and animal breeding was done for millennia before the theory of evolution came about by trial and error. But it is the theory of evolution that places plant and animal breeding on a proper scientific footing. The world map on the bottom left is summarizing what recent genetic studies of ancient DNA from skeletons from archaeological sites, etc., has told us about how anatomically modern humans or Homo sapiens sapiens moved around the world about 70,000 years ago. There were some earlier movements of modern humans from Africa to the the Middle East, and they seem to have died out without leaving a trace in the genetic makeup of today's global population. So today's entire global population of humans seems to trace back to one out of Africa event which started spreading humans starting from Africa around 70,000 years ago. This has been one of the great applications of evolution to understanding the period between where classical history begins, when you have civilizations and writing. And the positive message coming out of all this evolutionary genetic work on ancient DNA is that it tends to reinforce the overall understanding of prehistorical human migrations that had been developed by looking at linguistic evidence and back constructing linguistic phylogenies, by looking at historical, archaeological evidence, and so on. By and large, the evolutionary genetic data seem to support that understanding, and in some cases have filled in additional details. In particular, again, since we are in India, I would strongly recommend the popular book Early Indians that was published a couple of years ago by Tony Joseph, which summarizes the implications of a lot of this evolutionary genetic work in very easy to understand ways, and what it means for our understanding of how the Indian population came about over the millennia. I very, very strongly recommend that book. The last graph here on the right is what the data from genome-wide association studies, or GWAS look like. Now, genome-wide association studies are very popular nowadays to try to nail down genetic variants that are responsible for contributing to various biomedical conditions. And again, this is a very long story. It could be a seminar in its own right. I would just like to tell you that from an evolutionary genetics perspective, genome-wide association studies will most probably not work for any complex phenotype. And by and large, over the last 20 years that people have done these studies at millions and billions of dollars of expense, that is essentially what has come out of it. In short, nothing much has come out of it. And this was actually predicted on very sound theoretical grounds long ago by evolutionary genetics. It's just that so much money and so much prestige is now involved in these studies that nobody wants to admit that basically the whole approach is conceptually flawed and not. So listed here in this slide is a sampling of problems for which evolutionary biology really is what provides the foundation for possible solution. Multi-drug antibiotic resistance in bacteria. You all know that this is a huge problem today, and of course the antibiotic resistance is something that the bacteria evolved. This is an evolutionary problem, and it's going to require an evolutionary solution, or a solution rooted in evolutionary understanding. The construction of the solution will involve lots of things, including molecular biology, but the foundation of how you devise the strategy to deal with this problem is going to require evolutionary understanding. Another thing you read about a lot in the news is assessing the risks of genetically modified or transgenic organisms. Once again, producing a transgenic organism is an application of a fairly routine molecular genetic technique today. But assessing the risks of actually introducing those GMOs into nature is really a question in ecology and evolution, and you need that understanding and that insight to actually address this problem. Understanding the diverse mechanisms of aging and slowdown of aging in late life, I already talked about this a little bit. Trying to identify phenotypic, environmental, and genetic correlates of complex diseases. Complex diseases, or the so-called lifestyle diseases, are not going to be very amenable to molecular or cellular level biology research. They are going to require a much more ecological and evolutionary approach to be able to, for us to be able to manage. Using genomics to understand human history migrations, I just talked about it. This next point is very relevant given that we are in the middle of COVID, which is why I'm talking to you over the internet instead of enjoying being in Lucknow physically. How do new human pathogens arise by switching host species? The switching of hosts by parasites or indeed by symbionts is a classic evolutionary biology research question. And understanding the ecological situations that give rise to a higher probability of switching hosts by a pathogen is very important to understanding how these zoonotic pandemics arise. Another that brings together two burning issues of the time is to understand the epidemiological consequences of global climate change. This again is a hugely important thing. One very nasty and tragic outcome of this we saw in about a year ago with the outbreak of Nipah virus in Kerala is actually a classic case study in the epidemiological consequences of global climate change. How to optimize and manage genotype by environment interactions in crop breeding. Again, this is a classic evolutionary biology question. And to come to slightly more social level question, why is domestic violence asymmetric by gender? It's very interesting. Domestic violence is extremely asymmetric by gender. Also, the distribution of who kills who in murder cases is very asymmetric by gender. When a man kills another person, a huge proportion of the victims, if you take an average over a large population, are actually unrelated, that is not genetically related people. Often men kill other men. If you look at the much smaller set of murders committed by women, women almost always, when they kill, and they kill much less than men do, but they almost overwhelmingly tend to kill their close male relatives, often their spouses. Why are these asymmetries there? Believe it or not, this is actually a very interesting evolutionary question. Does monogamy benefit males and females equally? Very interesting evolutionary question, interesting in the human social context in understanding the evolution of patriarchal social systems, but also of interest in the context of lots and lots and lots of other species. The issue of whether monogamy versus polygamy benefits males and females equally is a very fundamental evolutionary question. Another interesting social question, unfortunately particularly poignant in India today, is rape primarily about sexual domination? So the question is, what is it that drives, on an average, what is it that tends to drive the crime of rape? The classical social science view is that rape is primarily about domination. It's not primarily about sex. Sex is secondary. The primary motivator and factor driving the committing of a rape is domination. This has been the classical sociological view for many decades. Evolutionary psychologists have argued that actually rape is probably primarily about sex and only secondarily about domination. And there is now quite a lot of data that would tend to support the evolutionary psychologist's view, or is at least very consistent with the evolutionary psychologist's view, whereas it is not consistent with the sociological view that rape is primarily about domination. Now, the reason this is of more than academic interest is that if we are to devise ways as a society of dealing with this problem of rape, it is important for us to understand what its primary drivers are and deal with them. And again, I'm not trying to suggest that what the evolutionary psychologists are saying that there are no socio-political drivers of rape. There are, but it is primarily not a crime of domination as much as it is about access to sex. And so far the data lean a little bit towards this explanation. How to use genetic data for forensic purposes and so on and so forth. So I hope what this part of the talk has done is help show you that almost all the big problems that fall into the domain of biology that we face as human societies, as individual societies, as a global society, almost all these problems are going to involve tremendous training and understanding in evolutionary biology if we are to come up with sensible solutions or management strategies for these problems. And this is a huge problem facing us because if you look at evolutionary biology in the Indian context, evolution is treated very, very rudimentarily or cursorily in school and university curricula. Much of the material we teach in our schools and colleges is wrong or out of date by decades. Can you imagine an MSc level curriculum in physics in any Indian university in which physics miraculously ended in 1900 before Einstein, before quantum mechanics, and before any of the other things that happened in the 20th century? Can you imagine a syllabus like that? And can you imagine the uproar that would happen if there was a syllabus? This is what happens to ecology and evolution a lot in our syllabus. It's like nothing happened after the neo-Darwinian synthesis. And this is a tragedy. It's also, we are doing an injustice to our biology students if we are teaching them nothing about ecology and evolution that happened after the 1930s. There is very little in the way we teach these subjects to suggest to students that evolutionary biology is a very active research field which has massive ramifications for multiple aspects of biology, agriculture, medicine, society. We give way too much emphasis to molecular biology. We tend to equate modern biology with molecular biology. And the unfortunate and uncomfortable truth is that for most of these major biomedical and societal problems that we talked about, molecular biology alone is not going to give us the solutions at all. We are going to use molecular tools eventually for producing some of the solutions, but the entire understanding, the entire foundational conceptual basis of coming up with strategies to deal with these societal and biomedical problems is going to come from evolution and ecology. It is not going to come from molecular biology. And it is high time in India that we got rid of this lopsidedness that we have in our biology education and research field. For example, in India, there is not one single institute at the national level devoted to evolution in its progress. Not one. We have a National Institute of Immunology. We have, I don't know how many institutes of molecular biology, cellular biology in various aspects. Not a single institute devoted to evolution. There's only one small department in my institution focused on research and training in evolutionary biology. And there was only one postgraduate training program in evolution, an integrated PhD in evolutionary and organismal biology in our department, which unfortunately was shut down because many people believed it was not really all that relevant. There is a huge irony in this neglect of evolutionary biology in the Indian context, and the irony is that in terms of fundamental contributions to biological knowledge and its growth, Indian evolutionary biologists have done far more than most other Indian biologists, including myself. In fact, I often keep hearing this phrase in meetings in Delhi and so on, sort of high-level meetings, where people say, where are the world leaders in biology research? And my answer always is, when you're looking in the wrong place. You won't find the world leaders if you look at the Indian molecular biologists. You will find the world leaders if you look at the ecologists and biologists. The second irony is that most evolutionary research is not very expensive and can easily be done even in less funded state universities or indeed undergraduate colleges. So the tragedy and the irony is that even state university and college faculty could do world-class research in evolution if they had a proper exposure to the field. if they had enough of an understanding of the field to be able to identify the interesting questions to work on, instead of being forced to do second or third rate molecular biology work because they don't have the resources, and they think that molecular biology is the only research worth doing. So they want to do only molecular biology, but they don't have the resources to do it. Whereas, if they had a proper education in evolution, they would be able to work on cutting edge questions, not requiring very fancy resources or expensive budgets, and would be able to do world-class research. So we are not playing to our strengths. In our wild rush to somehow we have to do molecular biology, we are not playing to our strengths at all. And especially for those of us who would like to do our evolutionary and ecological research in the wild, unlike me, I do it in the lab, but many people do it in the wild, we have another great strength, which is that we are a mega biodiversity country. And I think we should take to our strengths. So I'll now just give you a brief introduction to some of the fantastic work done over the decades from India and evolution. This is Professor Raghavendra Gadakkar at the Center for Ecological Sciences, Indian Institute of Science. This picture back at the bottom is Rhopalidia marginata, the primitively used social moth species on which he and his students did a lot of their work. This is his outstanding book, The Social Biology of Rhopalidia marginata, Harvard University Press. Professor Gadakkar did seminal work. There's this paper on the right hand side, it's a bit small, I'm sorry. I'll read out the title to you. It says, The Evolution of Human Sociality, the Advantage of Assured Fitness Returns. This was one of the major conceptual advances in understanding the evolution of sociality after Hamilton's work. This was published in the Philosophical Transactions of the Royal Society of London in 1990. Professor Gadakkar is the world's leading expert on the social biology of primitively used social hymenoptera. These are two remarkable people again, Umar Shankar and Ganeshaya, who their entire career was spent at the University of Agricultural Sciences, Bengaluru. Their mandate was to study crop physiology and plant breeding. But on the side, they also did pioneering studies of parent-offspring conflict in plants. Prior to Ganeshaya and Umar Shankar's work, parent-offspring conflict was only studied in animals. People didn't even think about it being a phenomenon in plants, and they really pioneered that line of work. They also rewrote the paradigm of one of the canonical textbook cases of mutualistic coevolution, the coevolution of pigs and pig wasps. And they literally rewrote the story on this one. Extremely, extremely important work, and very, very well recognized. This is Professor Vinit Vatve, also a great poet and musician and writer in Marathi. He was earlier at Garbhare College and subsequently for several years at IISER Pune. He is one of the few people in India who has done work in evolutionary medicine, in particular in developing evolutionary interpretations of type 2 diabetes, leading to a better integration of seemingly unexplained aspects of the disease. This is his book, came out a few years ago, Dove's Symptoms and Diabetes, a Darwinian interpretation of type 2 diabetes and related disorders. Now, the point is that this approach to diabetes leads to very novel suggestions, behavioral interventions rather than drug intervention. And some of these suggestions are now actually being tested in controlled clinical trials in the behavioral intervention for lifestyle disorders department established and functioning in the Dinanath Mangeshkar Hospital and Research Center. So this is promoting the testing. The trials are underway of behavioral interventions for dealing with metabolic syndrome and type 2 diabetes. And this comes from an evolutionary interpretation of what this disease really is and how it has come about. This is Professor N.G. Prasad, IISER Mohali. He works on fruit flies, uses experimental evolution, and is one of the top groups in the world. Is one of the top groups in the world studying interlocus sexual conflict, and they were the first to comprehensively experimentally validate in one common system all the many interlinked predictions coming from the theory of sexual selection and genomic level sexual conflict. They also provided the first experimental evidence of interlocus conflict being a driver of reproductive isolation and potentially therefore of speciation, and they were the first to start connecting immune system activity and the investment that individuals make in their immune system to trade-offs linking together lifespan and sexual conflict. And this is a field in which many other labs in the world have now followed the path that they first established. This is Professor Sudhir Day of IISER Pune. Among other things, he does many different kinds of work. I'm going to just focus on one kind of work they did. They did the first experimental evolution studies of adaptive evolution in multifactorially fluctuating environments. Usually, when you study evolution in the lab, you have a constant environment which poses some challenge. For example, if you remember the Dallinger study I talked about in my second or third slide, he basically faced his protists with a temperature stress every generation. So in Sudhir today's experiment, temperature, pH and various other factors randomly fluctuated from generation to generation, and they studied how bacteria faced with random fluctuations in pH and salt concentration in oxidative stress evolved adaptations via very, very different mechanisms than when they were challenged with one environmental stress at a time. And the most interesting finding from here was that these populations also evolved resistance to antibiotics they had never been exposed to, while they were adapting to fluctuating environments that were fluctuating in terms of pH, salt concentration and oxidative stress. They were not being challenged by antibiotics at all, and they had evolved resistance to antibiotics they had never been exposed to even in their past in the laboratory, and they were able to show that this was through the evolution of a generalized stress response to ATP-dependent efflux pump activity. This is a very interesting finding because another thing that people have been observing around the world in recent years is bacteria from completely pristine habitats like high altitude, pristine mountain streams, bacteria in the gut microbiome of completely isolated, primitively social tribes in the Amazon rainforest. All those bacteria tend to show genes conferring resistance to fourth and fifth generation antibiotics that they have never possibly encountered in their natural environment. And this suggests that antibiotic resistance can emerge as a byproduct of simply adapting to an environment that is fluctuating in many other environmental contexts. This slide is about a piece of work I am personally very thrilled about. This is a group of 4 of us, myself, Sudhir today and N.G. Prasad, whom you just saw, and my colleague at JNC and the chairman of our unit, chairperson of our unit, Professor T.N.C. Vidya. We have this informal association called Foundations of Genetics and Evolution Group, which we get together from time to time to work on foundational conceptual issues in evolution and genetics. Most of our areas of interest in this particular endeavor, which is not formal, not funded by anybody, this is just something we do because we really enjoy doing it. We are interested in reconceptualizing heredity in more inclusive manners rather than just genes, delineating the conceptual core of evolutionary theory, emphasizing newer ways of conceptualizing fitness and heredity together, and evaluating claims made by the proponents of the so-called extended evolutionary synthesis. And I'd just like to point out that this one paper mentioned here, Gupta et al. 2017, Niche Construction in Evolutionary Theory, the construction of an academic niche, is the first non-Western contribution to fundamental debates about the conceptual core of evolutionary theory since evolutionary biology in its modern sense began. And this 2017 paper is listed as being among the top 5% most discussed papers on the internet across all disciplines by our measures. This is a fundamental contribution to debates which one might say are about the very soul of the subject. This is not about adding one fact or 2 facts to knowledge. This is about debates about the soul of the subject, and by and large, Indian biologists have not been involved in such debates in the past. this is something that Dr. Gitanjali mentioned during her very kind introduction. This book was recently published, Conceptual Breakthroughs in Evolutionary Ecology, and the paper, 2003 paper is mentioned here. It was published in the Proceedings of the Royal Society, London, at that time, Biology Letters of Supplement. And this is the only non-Western study to be listed among the 65 breakthroughs, which begins with Darwin's theory of natural selection. And this is obviously something we are very happy about. And this is, of course, a picture of all of us as we look closer to the present day. When this study was done, N.G. Prasad was close to finishing his PhD. Sudeep Dey was just starting his PhD, and Mallikarjun Shakarat was a postdoc working with me at the time. All of them are now full professors. Mallikarjun is with University of Delhi Zoology Department, and Prasad and Sudeep are in another fact that people are often not aware of is that Indians are at the forefront of experimental evolution studies globally. If you look at all the labs in the world that use consistent long-term experimental evolution studies to address fundamental questions, more than half of them are actually in India. Can you imagine if we could honestly make the claim that half of all the labs in the world that study cancer biology are actually in India, the amount of media attention you will get? Nobody knows about this, but this is true. And experimental evolution in the modern sense began with the work of Larry Muller, who you see on the left here, and Michael Rose, who you see on the right here. And in India, experimental evolution began very early in the 1980s. Professor H.A. Ranganath pioneered the use of experimental evolution approaches in the Zoology Department of Mysore University. And as I said, we are very happy that actually India has a tremendous presence in the experimental evolution community worldwide. So to sum up and conclude, the need of the hour in India is one, the inclusion of a proper, detailed and up-to-date training in the concept and principles, not the details, but the concepts and principles of evolutionary biology at the school and college levels. A proper training in evolutionary biology for all biologists, not just those who are going to do evolutionary research. All biologists need a certain minimal level of proper training in evolutionary biology at the postgraduate level. And we really, really badly need at least one full-fledged national research institute for research and postgraduate training in all major areas of evolutionary biology, including human ecology. So I will stop here. Thank you very much for your patience, and I'll be happy to answer questions.
Thank you so much, Professor Joshi. That is actually a very relevant talk, and I really, really wanted our students to listen to this because, as you said, at the school level, most of the times, when they come to for undergrad and postgrad studies, they usually run away from evolution and ecology. They have this feeling in their mind. First of all, the main feeling is what is there to study in evolution? We know Darwinism, we know Neo-Darwinism, we know what Lamarck is. So they don't even bother to study it in much detail, though that is the general approach. And therefore, having teachers, I find that most of our students at undergraduate level are actually surprised to understand what survival of the fittest means.
Yes.
So then the very concept, they have cleared all their exams believing that it means that somebody who, it's an Olympics race with somebody who's running fastest or maybe jumping highest and has the most strength is the one. And the concept of offspring fitness and the number of offspring and direct and indirect fitness is completely lost in there. So it takes a good deal of effort to try and drag them to the side where they really begin to understand and appreciate the fact. So I think this talk has especially been useful, especially getting a perspective about how much is being done in India. That is very important for the students. So we do have some questions from the students. Yeah. So One is this is a research scholar who's saying that I agree with you on the fact that it is essential for us that the reason for negligence of evolution is probably because we have teachers who can hardly explain evolution in the classroom at the school as well as college level and are most comfortable leading it. And this neglected approach is also the root cause of cutting of funds. How do you think we need to address that? I mean, because it starts from the very fair. The people who are teaching are themselves not very comfortable with evolution.
Yes. So I completely agree with this characterization of the problem, and this is a classic catch-22 situation. We don't teach much about evolution, therefore we don't train people to be able to teach it when they become. So somewhere you have to break the cycle, and I mean, there are obviously many people will have many different views as to how this can be done. One is to actually, in addition to trying to undertake curricular reform, which is a very complex thing in a country, a diverse country like India, because education is also state subject or at least a concurrent subject, is to reach out to teachers and organize events where you get together teachers and you actually talk to them about how to teach basic concepts in evolution. You see, this is something which is largely lacking. What happens is, it is not that people don't do teacher training workshops. I have been a part or a resource person in many such workshops over the last 2 decades, organized by the Indian Academy of Sciences and by various staff colleges of various universities and so on. Usually what happens, you try to bring together a bunch of eminent scientists. Eminent scientists in general are often little bit in their own world. They come and give nice talks about their profound research. The poor college teachers really don't understand in any detail. And anyway, it's not relevant to what they have to teach. I started doing this long ago. I used to go to these teacher training workshops and, for example, give a 2-hour lecture on how to teach Hardy-Weinberg equilibrium without any algebra. And teachers universally said that they found it very helpful, because this is something many of them had to teach in their colleges, and they really didn't understand anything about it because they got scared by the equation. And the minute they saw that equation, they were like, oh my god, this is mathematics, this is not biology. How do I teach this? And my entire talk was basically saying, let's help you get rid of your fear of that equation, let's throw away the equation. You can explain Hardy-Weinberg just by talking about organisms and genes. You don't have to bring the equation into it at all. The equation is just a convenient way of summarizing what you can explain in words. And I'll show you how to explain it in words and what is its significance. So I think we need teacher training efforts which are designed to help the teachers how to teach, not just acquaint them with the latest research. That has its own importance, but we also need to help them with some of the basic concepts in evolution, some of the slightly mathematical concepts. How do you understand them and teach them without getting lost in the math or getting scared by the math? I think that is very important, and we need to do a lot more of it.
Yeah, true. A very new case of negligence of evolution in ecology was the chopping off the syllabus during syllabus reduction. that was another case of how beautifully, how much importance we give to these two subjects. I think a very important aspect is the way we disconnect. As you said in the very beginning, we consider evolution as a separate thing. Ecology is different, development biology is different, molecular biology is different. We do not realize that there is an interconnection, and in fact, everything that we are studying, as is very well quoted, nothing makes sense in biology without the light of evolution. Right? I think that perspective we need to give in everything that we teach. We need to tell them how it has evolved, and that is most important.
In fact, I am very thankful that you mentioned ecology, evolution and development together. I mean, I don't have time to elaborate on this. It's actually a whole talk by itself. But if you really look at the logical structure of biology as a discipline, in a certain sense, there are only three phenomena in biology: development, ecology, and heredity. And what connects these three phenomena is evolution. What evolution really does, evolution is how, or it's another name for how, ecology modifies development through the medium of heredity. And heredity need not be just by genes, it can be by non-genic mechanisms also. And by non-genic mechanisms, I don't just mean epigenetics, I mean cultural inheritance and ecological inheritance as well. So basically, ecology provides the challenge. In response, organisms, some do well, some don't. So some survive, their lineage survives, others don't. And they have different phenotypes, which means they have different developments. So the dominant form of development is change as a result of the ecological challenge. That is the sense in which ecology is changing development through the medium of heredity, which is what connects the generation. And that is all that evolution is, that is all that biology is. Everything else in biology is a detail, it's not a phenomenon. Now that is not to say that details cannot be important. Details can be very important. One little detail in development can mean the difference between your survival or your death as an infant. But they are conceptually, they are details. The three phenomena are development, ecology and heredity, which together constitute what evolution is.
Thank you. So there is a question by one of our students, Monish Singh.
Yeah.
He's saying, how can development be studied in the life of evolution? And so you've answered this. I'll go to the next one. He said, at the molecular level, the interaction of proteins is very intricate. How does phenotypic evolution relate to molecular evolution without making major deviations in the living system?
That's actually an interesting question. It's actually a question with philosophical sort of aspects to it. See, of course, at the level of the molecular things that are happening within a cell, or in some cases between cells as well, things are quite complex. But organismal phenotypes are a very different beast altogether. And the point is, see, we don't need to understand just organismal phenotypes, we need to understand differences in organismal phenotypes, because that is what natural selection then acts on. Now, differences in organismal phenotypes do not, by and large, there are exceptions, but by and large, they do not arise due to fundamental differences what is happening at the protein level in the cell. At the level of what is the cellular mechanisms, all organisms are the same. Every organism mitochondrial functions exactly the same way. So this individual different from that individual, the molecular level is not where you are going to find the difference. It's at the organism. And a simple example of this is, when you go to buy a car, what do you look at? You look at its appearance, its color, its design, its comfort, its mileage, its stability, its luggage space. You don't look into the chemistry of how petrol burns in the engine. You don't look into the physics of how the differential transmission shaft works. Because how you will find which model of car is suited for you doesn't matter at the physics and chemistry level. All cars are the same. All car engines work in the same way. It really doesn't matter. What you need to evaluate is the organismal level phenotype of the car and find out which one suits you. That is what natural selection does. It looks at the organismal level phenotype, figures out by selective killing or reduction in reproduction which organismal phenotype is not working in that environment.
That was a very nice simile, cars and this thing. I'm going to use that in class next time. Thank you for that. Yeah, so one more question, which is that in general, if we look at evolution as a whole, evolution has been found to move towards complexity, right? I mean, if we look at
Actually, I'm going to disagree with that as well.
Oh, okay. Yeah, so that is what the question was. It's by a student who says, if evolution we assume moves towards increasing complexity, then why do simpler organisms work maybe better or finer in certain aspects?
Absolutely. So first of all, in some lineages, if you take one little branch of the evolutionary tree or the tree of life, you might be able to find a few small lineages where it looks like there is the evolution of increasing complexity. There is no such general tendency across the entire evolutionary tree at all. The second thing is that even in individual lineages where there seems to be a tendency towards increased complexity, that's an empirical fact. But it doesn't follow that there has been selection favoring increased complexity there. That's just a speculation. In many cases, the increased complexity could be a byproduct of selection acting on a different set of traits. But overall, in the entire tree of life, there is no such tendency towards increase. In many lineages, you will find the evolution of decreased complexity. In many parasite lineages, quite well.
Thank you so much, Professor Joshi. It's been a real pleasure. I was actually looking forward to this talk with great excitement, and I was telling the students also that they really need to hear it. And our MSc third semester students have actually got evolution this semester as a part of the course. So I think especially for them, it's been very good. I'll just hand over the mic to our head of department, Professor Sudhir Kumar. Sir, if you could just unmute yourself and say a few words. Yes.
Thanks, Professor Joshi, to be with us in this beautiful morning. I think, as you rightly said, that it is time for the evolution in the Indian universities. And as J. Muller proclaimed at US that 100 years without Darwin is enough. So we can say that now the time of reduction biology is over now. We have to move towards the system biology. And as you rightly said, because I'm a student of evolution also and get the opportunity to teach at the BSc and the MSc level, I think the conception of origin of life is the main hurdle. The fear and the earlier knowledge of the science or religion is the main handicap in the evolutionary research and the evolutionary understanding. It's a beautiful thing that your lab and this many institutions, as you rightly enumerated, are doing exceptional work. But in the northern Indian universities are poor in that respect, that we are not undertaking as much. I think we have missed this opportunity, lockdown opportunity. We could develop more, but we could be involved in the beautiful studies at that time on the evolutionary, social evolution or the behavioral evolution. But I don't know whether any lab is doing that. Thank you to be with us, and I think Gitanjali and Sally did marvelous job in this organizing this talk. And I'll be fortunate enough if you visit our department. I will invite you for some physical interaction because our students are excited. I'll invite you. Thank you.
I would definitely, once this whole pandemic situation subsides, I would be very happy to visit in person. You know, I mean, especially interacting with students face to face is much more fun.
So
Absolutely.
Let me say you're most welcome indeed. I should thank the organizers and Dr. Gitanjali in particular for giving me this opportunity. It's a great pleasure for me to speak, and that too on such an august occasion like a centenary celebration of one of our great universities of my home state. So thank you very much. It was a great pleasure for me.
It is our pride also, sir, that you belongs to the UP, and we are waiting for you here. Thank you, sir.
Thank you, Professor Joshi. Thank you so much.
Yes.